The next one's phosphorus cycle. Phosphorus is extraordinarily important because it is the limiting or a co-limiting nutrient in many situations, a key ingredient to eutrophication problems. So understanding the cycling of phosphorus is essential to understanding how humans are affecting aquatic environments. That said, there's not a gaseous form that's important. There's not a bunch of intermediate forms that are important. And so we really think about three forms: organic phosphorus, phosphate, and then it turns out it will also precipitate with iron. So iron phosphate is an important precipitate. So I don't make you memorize a cycle because it's fairly simple. There is a diagram in the book that does give you a simple cycle if the pictorial version is easier for you, if the cartoon is easier for you to memorize, then you can use that to guide your process. All organisms need to take up phosphorus. They can either do it from the inorganic form, if bacteria and plants and such can use phosphate directly, or as heterotrophs with the inability to use simple inorganic compounds, we have to take it up in the form of lipids, nucleic acids, and those types of forms. We also have continuous mineralization, and for this mineralization, phosphatases are extraordinarily important. Basically things that cleave phosphate off of organic molecules. They're also central to the way our bodies work, right? Because we do ATP to ADP, that takes a phosphatase. Remember, the microsystems are nasty toxins because they interfere with the activity of phosphatases. So it's already showed up once in our class. And finally, precipitation with oxidized iron. We'll talk about that in a bit. Silicon, so essentially every trace element has a cycle, whether you talk about iron, manganese, anything that organisms use biologically has its own cycle. But I've not talked about those in much detail because that's not the point of this class. So I've tried to do is pick out the cycles that are important to the general functioning of aquatic systems and understanding them at the most basic level. And then hopefully you'll see how those link into, if you're not interested in the cycles in their own right, water quality or fish production, eutrophication problems, those kinds of things. The silicon cycle is important predominantly because it's a key element in diatom frustules. And diatoms are central primary producers in aquatic systems. At times it can become limiting in lakes and also in rivers and oceans. It can change things. So we have here an annual cycle of silicon, and this is from a series of about 50 years in Lake Windermere in England. And we see that every spring the diatom phytoplankton populations go way up, and we'll talk about seasonal succession of phytoplankton and why this might be. But as these go up, the silica concentrations drop, dissolved in the water, and then as the bloom goes down and the cells sink out, and they start redissolving back into the water column slowly, the silica comes up and down. It's this repeated cycle over the years. So you can see how it could be limiting. This could be part of the reason that these populations drop as a central nutrient to them. So one of those graphs where you have time throughout a year on the x-axis, and the y-axis is depth from surface to 12 meters and surface to 5 meters. And this is a very eutrophic lake here, and this is an oligotrophic lake here, and the times aren't exactly the same, if you'll notice. So winter happens to be right here. And what we see is that it's a very low concentration of silica in the winter and spring when you have those blooms of diatoms. And then as the lake stratifies in the summertime and those diatoms settle out of the plankton, the silica starts to dissolve in the late summer at the bottom. So there's also the spatial distribution of silicate. And we have pretty much the same situation where we have this ice off, we have this production in this oligotrophic lake of diatoms in the summertime, and then it falls down, and by fall it starts building up, the concentration starts building up in the bottom. We can also see some relationships like this in rivers and streams. So one of the things that has changed in the Mississippi River is that the silica levels in the Mississippi have gone down. And that's because nutrient additions from cropland have increased the amount of nitrogen and phosphorus, which has stimulated the diatoms that take up the silica that are attached in the river systems coming into it. That means less silica is going out into the Gulf of Mexico, and the nutrients are stimulating growth of algae in its forms that are not as easily grazed. So there's this cascading effect that does involve the silicon cycle as well. We haven't talked about this much, but there's a dead zone that forms in the Gulf of Mexico, where the nutrients from the Midwest go out into the Gulf of Mexico, fertilize the phytoplankton, they sink down, and then as they're decomposed, the oxygen gets used up, and there's this low oxygen zone, dead zone, that migrates around in the northern part of the Gulf of Mexico and has really big negative impacts on the fisheries there. The fish can swim out of it, but a lot of but some of them get trapped, and also the shellfish tend not to do so well in that. So it has big effects on the fishery, and it's a major concern. But in addition to that fertilization, it's also shifting the stoichiometry. So there's a move towards less diatom production and other algae being the primary producers. The other algae are less easily grazed by the food web there, so it's disrupting the food web. Even though there's more production, it's not necessarily making it into the food web. So that's a sort of a segue into this idea of interactions of nutrient cycles, and that's the major point of this part of the lecture that we're talking about. And the last of the cycles we'll talk about in detail before we link them is the iron cycle. And we have two forms of iron, the oxidized form, ferric, Fe3+, and the ferrous, the reduced form, Fe2+. Ferric iron is actually not very soluble. And so it precipitates in solution, forming this rusty sort of stuff. And we talked about when you have groundwater from agricultural areas, that I'm up 2 slides or so. We have this agricultural, in these wells that are low oxygen, that have high iron in them. When you pull the water up and you oxygenate it, then the rust forms, the precipitate comes out of solution. That also happens in lakes, because when you have these high iron that builds up in the anoxic hypolimnion, and when it mixes up into the epilimnion in the fall, when it mixes up through the whole surface, then it'll precipitate out and fall down out of solution. Fe2+, though, it spontaneously converts to Fe3+ when it goes from reduced to oxidized conditions. If the microbes can grab it before it is oxidized abiotically, the chemoautotrophs can generate energy. So it's just like sulfide, right? When sulfide comes into contact with oxygen, it will convert to sulfate abiotically. But if the organisms can get that sulfide and the oxygen and combine them, the chemoautotrophs can create energy from that. The same with reduced iron. When reduced iron comes into solution, it starts converting to Fe3+ spontaneously and then precipitates out. But if the microbes can get that reduced iron and oxidize it, they can gain energy off of it. And so this leads to formation of nodules or deposits of iron in the bottom of some lakes, and actually in the bottom of the ocean. There's these, you may have heard about mining the nodules of metal that are in the bottom of the ocean, and these are formed as reduced iron comes out. Again, in some of the lakes, particularly further north from here, that occurs as well. One place where you might have seen a lot of iron formation in a stream nearby here is at the small streams that drain the bottom of Tuttle Creek Reservoir, and they're very reddish color. I don't know if you've noticed it. There's a reddish precipitate all over everything there. That's where they throw the trout in. If you want to get a marshmallow and put it on your hook, you can catch one of those trouts. It's a highly natural situation, but if you ever go there, how many of you guys have seen those little streams there and noticed how orange they are? Any of you seen that before? Got a few of you. That's because the low oxygen water from underneath that dam is coming out from the drainage system, and it picks up iron, and when it hits the oxygen, it starts precipitating out. And there's actually good bacterial growth there that's stimulated. Those chemoautotrophs are stimulated by that. So now next time you go by the bottom of Tuttle Creek Reservoir, maybe you're out at Country Stampede or something, you can wow your friends with all those fantastic bacteria that are growing there. The one thing that this Fe3+ will also react with is phosphate, and so if you're pushing phosphate into the environment and iron together into an oxidized environment, they will form an insoluble precipitate that will settle out. If that settles out into an anoxic zone, then the iron will convert back to 2+ and release the phosphate. This is quite important because this precipitate, if you have oxygen in the hypolimnion, this precipitate then goes down and sits on top of the sediment and locks up the phosphorus. But if you push a lake to the point where the hypolimnion becomes anoxic, the phosphorus will settle out and then release and then come back up into the solution. And we'll talk about this when we come to eutrophication. It's a key factor for why you take a lake, and if you push it past the trophic state where you lose all the oxygen in the hypolimnion, you enter into a very difficult state to reverse. It becomes very difficult to reverse eutrophication if you allow this iron phosphate to redissociate into the environment. Okay, so we're starting to see now, I've given you a couple examples of how things link together, of how nitrogen and phosphorus pollution can change the silicon ratio, of how iron can also react with phosphorus. And then, remember, there's one more thing that iron can do. One other thing that iron can precipitate with. You remember that from the last major cycle we talked about? Sulfur, sulfide, right? Iron pyrite can form. It makes an insoluble precipitate in anoxic conditions. And so we can actually link the sulfur and the phosphorus cycles. And so understanding these cycles then allows us to manage aquatic ecosystems better. And this brings me to one of my favorite examples that I often ask on exams, some variant of this particular type of question. And students often don't understand it because it's quite complex, but it means that if you can figure this out, why this worked this way, you understand the complex linkages, some of the complex linkages between the nutrient cycles. They're clearly linked to each other because all the biota uses all the major nutrients, right? But there are also other chemical interactions that make these happen. So this is an example of phosphorus pollution in the lowlands of Netherlands. Encouraged unwanted algal blooms, and they were actually trying to protect the native vegetation of the wetlands, the macrophytes. And so what they did is they decided that they needed to get rid of this phosphorus pollution by stripping phosphorus out of river water and flooding and flooding the wetland with that, increasing the throughput of low phosphorus water. They built a plant to strip the phosphorus out chemically, and actually they use iron to strip the phosphorus out. They'll precipitate it out and then and off it goes. But when they put this water in, it had high sulfate. What turned out happening is the sulfate was reduced to sulfide. Okay, what process reduces to sulfate to sulfide? It's analogous to respiration, right? But it happens in the absence of oxygen. That's dissimilatory reduction of sulfate, right? Remember? So microbes can use sulfate to oxidize organic carbon, right? They can use it to accept electrons as they oxidize organic carbon, with sulfide as the byproduct. And oxygen, using oxygen, aerobic respiration is the most efficient. Using nitrate, denitrification is the next most efficient. Using sulfate is the last one, and then once that sulfate goes away, then you convert over to methanogenesis, being energetically favorable. So this sulfate came in, there's plenty of organic carbon in the sediments, and it's converted to sulfide. The sulfide then made pyrite, got rid of all the iron, and then the sulfide levels built up, and sulfide's a quite toxic molecule. It was poisoning the macrophytes, so it's taking them out. When the iron was not there to bind the phosphates that were in the sediments, the phosphorus actually came up in the wetland. So essentially, they exacerbated their problem. They put in low phosphorus water, but because the sulfur was high, they poisoned the plants out that they wanted to keep. The sulfur dropped the iron out, and they increased the rate of phosphorus input into the wetland. Basically, a waste of money because they didn't understand the interactions between the nutrient cycles. So that's one that I think is worth thinking about, because if you really understand the way this works, you understand linkages among the nutrient cycles. And I know it's complex, but it's good for you. Put your chemistry to work. Okay, so we'll talk about this in a more general sense, and we'll try to link these all together in a series of diagrams here. And this diagram could be depth in centimeters into a sediment. It could be depth in meters in a eutrophic lake, right? And we go from having fully saturated oxygen to anoxic at some depth. We'll just say it's in the sediment. And the redox goes from being quite high to negative with depth. So this is exactly what I was talking about before. This oxidized area can have iron phosphate dissolved in it, right? I mean, iron phosphate precipitated in it. But if you go to the anoxic, the Fe3 plus is converted to Fe2 plus, right? And that no longer binds to the phosphate. So this oxidized layer is trapping that phosphorus in there because it gets stuck with the iron. And we can play the game with all of the different elements. So here's the pyrite forming anoxically, sulfide anoxically. When it comes into contact with the oxygen, either abiotically or biotically, it is converted to sulfate, right? So that is either an abiotic or a biotic chemoautotrophic reaction. And the sulfate is also being converted to sulfide as it comes down to these anoxic areas because it's being used to oxidize organic carbon. So we have sulfate reducers down here and the sulfur oxidizers here. The chemoautotrophs that are oxidizing sulfur up here where sulfide and oxygen are together. Where there's no oxygen, sulfate can be used as an electron acceptor to oxidize organic carbon. The sulfate reducers are producing sulfide from sulfate. And we can look at what's happening with the nitrogen. Right? So when we're anoxic, the ammonium's high and the nitrate's low. Because the denitrifiers are doing the same thing as the sulfate reducers are. They're using nitrate as an electron acceptor to oxidize organic carbon, and some of them are giving off ammonium, right? Or it's going off as nitrogen gas, and mineralization is increasing ammonium. as the proteins break down. As this ammonium diffuses out into the oxygen-containing part of the sediments, nitrifiers can oxidize that ammonium chemoautotrophically to make energy. And we can look at the iron. Reduced iron down here, oxidized iron up here, iron oxidizing bacteria sitting right here. Fe3 plus is actually precipitating with FeOH, right, and then making this precipitate down here. And the iron oxidizes as it comes out, have this high population. And we can look at the methane. Methane's high in the very low redox areas, and that's because it's being produced by methanogenic organisms. These methanogenic organisms occur below the sulfate reducers, right? Because you have to use up the sulfate first, and then you get low enough redox that you'll get methane production. And that methane is diffusing out, and the methanotrophs, the organisms that eat it, are oxidizing. What this says is that the anoxic-oxic interface is a biogeochemical hotspot. It's where a lot of the action is occurring in most ecosystems. And I told you I'd go back to this figure that we had a ways back, and it was all the way back in the carbon. But I promised you I'd go back to it. It's too far. There. Okay. So I'm hoping that this actually makes a little bit more sense to you now that we've talked about it, because it's the same sort of general thing as I was talking about. It only has the oxidizing stuff on it, but it sort of has half of it. But again, we go down with depth. We have oxygen at the surface, and the redox is getting lower and lower as we go down deeper. If we have organic carbon up here, it is being converted to CO2 using oxygen as an electron acceptor. That has the highest energy yield of the oxidations, the biological oxidations. As we move down to the sediment and the oxygen disappears, nitrate becomes the preferred electron acceptor for oxidizing organic carbon. You're generating into, or under very low redox conditions, you can go to ammonium with dissimilatory reduction of nitrate to ammonium and producing CO2. I didn't talk about it, but oxidized manganese can also serve as an electron acceptor, as can oxidized iron. You don't have to remember those if you don't want to for this class. But you do have to remember that sulfate can be used as an oxidized, an electron acceptor to oxidize organic carbon, giving rise to sulfide. And that once that sulfate's gone, then the redox gets driven low enough that methanogenesis, using CO2 or acetate to produce methane, can actually increase the rate of energy production for these organisms. And